congratulate the organizing committee for the tremendous evolution of this meeting, ALPIC. You know that there has been a tremendous evolution, and what we see here, distinguished speakers, among them Lina Badimon and Dr. Veyron from Israel, Professor Catanao from Italy, are coming to us because the meeting has become very prestigious. And for me, Lina Badimon was a well-known name since the 90s, when I read much literature, and I saw many works together done from you and Professor Cesebro.
Yes.
So I think you have made a remarkable career in the field of hemostasis and thrombosis. And for the first time, I'm very, very pleased after 30 years to be able to sit together with you, co-chair with you, and introduce you. You are chair in Barcelona and in cardiovascular research in Barcelona, Spain. And she has, I can tell you, more than 500 publications, very important publications on this field. In many of them, she's a first author. She's a pioneer on the field, and I'm glad to introduce her to you. Lina.
Thank you very much for this nice introduction. Today I'm going to speak about tissue factor signaling, and I'm going to speak about issues with tissue factor that we are still discovering. Tissue factor is known for many years, but my title goes beyond coagulation in the cardiovascular system. So this is the coagulation cascade, something that is common to all of us. And tissue factor is sitting there in the extrinsic pathway and helps to trigger what is called vascular wall-induced thrombosis. And in the 90s, as George said, we were very much interested in understanding how this thrombosis in association to atherosclerosis was being produced. Nowadays, the learning of how tissue factor works at the level of triggering thrombosis has produced many different options for treatment. And you are going to be listening to those today, and coming like it happens always in medicine when we have new compounds. Now, these direct factor 10A inhibitors are going to be used also at the arterial side, because the arterial side has also a lot of thrombin formation and fibrin formation eventually to form these thrombi. Therefore, we are evolving with time and going a little bit trying to find better drugs to inhibit thrombosis. Yes, in the 90s, we really wanted to know how the plaque was able to trigger these thrombi that were able to occlude the vessel wall. And we found that where the inflammatory cells within the matrix of these atherosclerotic plaques, those compounds that were able to trigger formation of this fibrin mesh that was evolving from tissue factor triggering of the coagulation cascade. And we saw that obviously it was factor 7A was involved, and what we know about the coagulation cascade. And when we did some studies with different types of atherosclerotic plaque back in the 90s, we saw that they were the lipid-rich core plaques, those that were very rich in inflammatory cells and lipids, those that were able to produce the higher level of platelet deposition. So although tissue factor triggers the formation of fibrin, platelets reacted equally, and the thrombi were very rich in platelets. And this was associated with these lipid-rich cores and with the presence of tissue factor. Those plaques richer in tissue factor were those that were able to trigger the larger size of thrombi. Now, we were able to demonstrate that this was tissue factor dependent because of the TFPI blockade of this phenomena on the top of atherosclerotic plaques. Well, the tissue factor molecule, as I say, is complex. is a cytokine family receptor that has, in addition of linking to factor 7 and triggering coagulation, has many other different functions. And we are trying to really identify those. In our hands, we have been trying to understand why tissue factor was sitting sometimes in the media of atherosclerotic plaques, never present in healthy media, but yes, when atherosclerotic plaques was growing, and it was not associated to macrophages. So we didn't know what it meant. Parallel to those studies, we were identifying those plaques that had the tendency to rupture in the coronary arteries, and saw that those plaques were rich in microvessels, new vessels that were growing in the thickness of the atherosclerotic plaque. This is an example of plaques obtained from patients that had heart transplant in our institution, and you see that those plaques that were classified following the American Heart Association as plaques type 5 were those that had highest number of microvessels. The same results by the same years were found by others, as you will see. Now, all these microvessels that grow in the mass of atherosclerotic plaque are not equal. There are some that are really competent. They have pericytes around the monolayer of endothelial cells, and they are not able to break as easily as those others that are only endothelialized, meaning that they are friable and able to rupture, producing severe hemorrhages within the atherosclerotic plaque. And are those hemorrhages within the atherosclerotic plaque those that trigger all the inflammatory process and rupture? And here you have the examples of these two types of microvessels. These ones are microvessels that did not produce any hemorrhage, and these you have in the same plaque in other side, those that produce hemorrhage. This atherosclerotic plaque comes from a patient who died from sudden death. So we don't have too much information, but other groups were looking into the same thing. And by the same years, this was in the beginning of this century, the group of Renu Virmani also was paying attention to these microvessels, and they were able, by sophisticated technology, to see them penetrating the plaque from the adventitia, and they saw that they were associated, the presence was associated with plaque rupture. And the same, this other group, where they found that the association of the presence of microvessels was with acute coronary syndrome patients. Therefore, these microvessels produce instability in the plaques. Now, what have we done? Looking into tissue factor, because we have been very much interested in tissue factor for all these years, we set in culture microvascular endothelial cells. And we saw that the tissue factor seen in green in these two slides, although maybe we can turn the lights, well, there is nobody, the lights on the front. Can we turn down the lights on the front? So you see in green tissue factor. In static cells, tissue factor is in the Golgi, and these cells are not having activity, but when you induce them with serum to migrate, tissue factor is moving around the cell surface. So we saw the cells were able to orient tissue factor in different positions. And we thought that maybe it was going to be very interesting to see if we could associate tissue factor with this microvessel formation. As you see here, this was published already 8 years ago, was the beginning of our effort in our group to understand this phenomena that it's very clear if we look into the images. When we co-culture smooth muscle cells and endothelial cells in normal condition, they produce these vessels in the collagen plates. And this is a macroscopical observation of one of these tubes. You see that endothelial cells are red and smooth muscle cells are green. And they acquire this typical position of the cells, one inside of the other. However, when we silence by molecular techniques and genetic techniques tissue factor in the macrovascular endothelial cells, there is no net formation. When we instead block tissue factor in smooth muscle cells, the endothelial cells that have tissue factor try to organize vessels, but they are not followed by the smooth muscle cells. When we block, by blocking the gene tissue factor both in endothelial cells and vascular smooth muscle cells, there is nothing. Cells don't move. And when we overexpress tissue factor in endothelial cells, we have this mess of angiotubes that forms because we have an excess of tissue factor. These studies in vitro led us to continue investigating how tissue factor was able to organize this growth of microvessels. So we did it in vivo. And here you see the same results. These are microtumors that are placed in the dorsal aspect of mice that are immunodepressed. Therefore, they cannot react to human tissue factor. And here you see the situation with wild type. So you see vessels form. When we silence tissue factor, nothing. When we silence tissue factor only in endothelial cells, there are hemorrhages because the cells try to organize themselves, but they cannot. When we overexpress tissue factor, there are these beautiful new vessels. There are significantly much more number of vessels than even in normal conditions. And here is when we don't implant any cell. Again, we have an in vivo demonstration that tissue factor expressed in endothelial cells is able to form neovessels. This was really very interesting for us. And through different studies, we end up with the signaling mechanism and machinery that the endothelial cells were using to produce this network of vessels. And this is how it happens. Tissue factor in the surface is stimulated, signaling is produced through AKT, ERK, and ETS1. ETS1 is a transcription factor that is able to recognize the element in the DNA that recognizes ET1 and triggers CCL2. CCL2 is released and is recognized by CCR2 in smooth muscle cells that are brought to the vicinity of the endothelial cells. So we had discovered how these endothelial cells are able to attract these other cells that are going to produce stability. If this is interrupted, the neovessels that are formed are not able to sustain pressure and they break when blood circulates. Therefore, we had here the first step in our research. We need to organize vessels in atherosclerotic plaques that are rich in tissue factor and produce stable vessels that will not break. And then the plaque will not be a high-risk plaque with a tendency for rupture. Next, we wanted to see that this effect of tissue factor was not dependent on BGF. I'm sure you have heard a lot of BGF and the BGF capability to form angiogenesis. You see here that when we had silenced tissue factor, BGF was not able to rescue the phenotype. Therefore, you see it here. This is control endothelial cells when tissue factor is blocked, and this when tissue factor is blocked, and we add BGF. So it's not dependent on BGF. It's an effect derived from tissue factor in itself. So we went into plaques and said, well, this is very nice, but maybe this is something that we are finding just in tissue, in vitro, and in rodents. Let's see if we have this in human plaques. And we went into our repository of human plaques, and you see here the results. If we look into this area here, this is a normal vessel. This is a vessel, a human vessel with a significant plaque, and we went to study into the shoulder of the plaque, where usually the rupture of the plaque starts. And these are magnifications. Here you see that we had staining for von Willebrand factor, and we saw not only the luminal endothelium, but also small vessels in the inside, tissue factor, staining only the internal vessels, not the luminal endothelium, and by fusion of both, the merging, we see in yellow that are these vessels within the atherosclerotic plaque, those that are positive. And to be really clear, is this other slide that magnifies that. So there is no tissue factor stain in luminal endothelial cells. And that was important for us to find, because we thought, well, if tissue factor is in luminal endothelial cells, how we explain that there is not always this prothrombotic phenotype. Therefore, there is heterogeneity of endothelial cells, and there are some that are able to express tissue factors and others that don't. And this is something that remains to be investigated. We don't know how this is processed as yet. But the fact is that these human plaques have tissue factor-rich microvessels, and these tissue factor-rich microvessels are important if they contain another type of cells of the type of pericytes of smooth muscle cells that make them stable. So we did to understand the mechanism, silencing or overexpressing tissue factor in these microvascular endothelial cells, and we have hints of different genes that are upregulated or downregulated. So we have a lot of work in the future, but in reality, what we are seeing is that tissue factor is able to control many different other genes. And we are not talking here of coagulation. We are, because in the system we use, coagulation is absent. Therefore, tissue factor is a really important molecule for different cell functions. Then we analyze if these cells that are rich in tissue factor are able to secrete microvesicles that contain tissue factor. And this is the fact. You see, these are quiescent cells, migrating cells, and angiogenic cells. We saw that these angiogenic cells were able to secrete microvesicles. And then we analyze the content. If we look into this last column, we have these are quiescent cells, these are migrating cells, these are angiogenic cells, and this is the content on tissue factor. So the angiogenic cells are able to secrete larger amounts of tissue factor with these microvesicles. What's the function of this secretion? Well, first we wanted to see that these microvesicles were received by other cells, and if by crossing with other cells, we could have distal signaling. And we saw that indeed they are able to link to other neighboring cells, as you see here attached. These are in vitro experiments where you clean the supernatant, so what is left there is attached. So these microvesicles were able to attach. And by a different type of setting, what we look at, it was this microvesicle able to trigger migration from aorta of cells. And you have it here. This is the control, VGF. This is control, blank control. When the microvesicles were rich in tissue factor, this is external tissue factor, bot tissue factor. When we block B1, I didn't show you because there's a lot of biochemical data, but we identified the mechanism of crosstalk between these microvesicles and the receptor cells is B1 integrin dependent. And here you see it is the case. Here is another control with VGF, and this is when these TF positive microvesicles are confronted with an antibody anti-V1. This is the parallel with the blockade. So we have an effect that is microvesicles rich in tissue factor trigger migration of endothelial cells and is a mechanism dependent on B1. Is it true in vivo? It is. Again, the demonstration that this is tissue factor rich microvesicles, and you see that these microvesicles are able to trigger this formation of vascularization in the skin of rodents. But the most important result is here. Does it have any functional capabilities? And this is something we were pursuing for many years. And here you see, by doing, this is MRI, what you see white means that blood is circulating. So it's not that a tube is formed, but blood is passing through these vessels. This is a model of heme limb ischemia. These are rats in which we have tight circulation in the limb. Then is when they are exposed to tissue factor negative microvesicles, and this is when they are exposed to tissue factor rich microvesicles. As you see here, not only is the fact that we have microcirculation, we have collateral blood formation. And if you see here, this is a transversal cut of the machine, the MRI. You see these are when the microvesicles don't have tissue factor, and this is when they contain tissue factor. So the formation of these new vessels is an important issue that we have already demonstrated in a porcine model. I don't have the data with me, but when we do the treatment with tissue factor into the heart of the porcine model, the model that is closer to our system, and that we intervene by using balloons, we saw that tissue factor was able to reestablish or induce the formation of collateral vessels. This is something that is really most important in our view, But we have mechanistic questions still that are not answered. So how were microvascular endothelial cells initiating the TF upregulation machinery? Because in vitro we could induce them. But what in vivo can induce these microvascular endothelial cells to produce tissue factor? And which were the triggers? So we have done research to understand all this process, and we found out that these monocytes were the cells that induce tissue factor. And it's clearly seen here. These are control situations. This is the skin. This is the tumor. These are microvascular endothelial cells plus monocytes. So this is the condition media that we present here. And you have a certain level of vessel formation. When we silence Wnt5, Wnt5 is a product secreted by monocytes, we have an inhibition, clear inhibition. And when we have this silent Wnt5, but we put external Wnt5, we recover function. And you see here by histograms the significance of these changes. Now, this is the model that we have already with different experiments. Monocytes secrete Wnt5. Wnt5 is recognized by Frizzle5 in the membrane of endothelial cells, and this Frizzle5 triggers the Wnt signaling pathway, go to the reticulum endoplasmic, regulates calcium secretion, activates NF-kappa beta, and NF-kappa beta, being an inflammatory transcription factor, triggers tissue factor secretion. Therefore, we have a link that is trying to tell us that this is possibly happening when we have this collateral vessel formation. But then, what's the source of these collateral vessels? Is only endothelial cells that duplicate themselves to form the new vessels, or there are other cells that are participating? Can be that monocytes differentiate into endothelial cell-like to produce this vessel. And this was the hypothesis we wanted to test. We know this pathway. We know monocytes trigger Wnt5. Wnt5 triggers angiogenesis because of tissue factor secretion. And endothelial cells also upregulate tissue factor and can form angiogenesis. But is it possible that there is transdifferentiation between monocytes and endothelial cells? It will be a new source of cells to produce this new vessel. And this is the case. These are cultures of monocytes, CD16 positive monocytes with endothelial cells. When we block a tissue factor and when we overexpress tissue factor. And you see clearly that the phenotype of the cells here are acquiring this spindle shape that are producing markers of endothelial cells. So there were no endothelial cells to start with, but with the supernatant of those cells are able to produce endothelial cells. And here you have that these cells, in presence of tissue factor, express cadherin, von Willebrand factor, and enos, 3 markers of endothelial cells. So we believe that there is the differentiation of these monocytes. And we went to the in vivo model when we have this collateral vessel formation. And you see in red, some of these new vessels, these in red, are monocytes that were marked with a light fluorescent color red, and you can find them forming the continuity of the vessel. More research is needed in this area because we have to know when and how these monocytes get dedifferentiated into endothelial cells. But possibly this is something that is happening at the level of atherosclerotic plaque. So this is our final diagram at this moment. So the monocytes that are activated can release Wnt5A, recognized by Frizzle5, trigger signaling through calcium and NF-kappa beta in the cell to release more tissue factor. And this tissue factor, the full length or the alternative splice behaves similarly, are able to really regulate back these monocytes to initiate transdifferentiation into endothelial cells. And simultaneously, these endothelial cells, by the mechanism I showed you at the beginning, are able to contribute to angiogenesis. So there is a lot to investigate as yet, but I think we are now going to work not with the full length or the alternative splice tissue factor. We have mutated the tissue factor molecule. So, because this is an effect that is important in many conditions to stimulate ischemic tissue or repair ischemic tissue, we have to make sure that we block the procoagulant part of tissue factor. And that's what we have done by mutating the areas by which tissue factor links to factor 7. This is mutated here, and we have mutated other different amino acids, and we have segments of tissue factor that are triggering angiogenesis, and in which we are going to investigate further to be able to associate function to the molecule in itself. Thank you very much for your attention. I want to thank the collaborators.
Thank you very much, Professor Badimon. It was an exciting lecture on a tremendous amount of research, which I must say it was for me very difficult to follow, and I made many efforts to keep the main points. And before I ask you, I will ask the audience if they have a short question to get a short answer. Everything has understood everything. Everyone has understood everything. Dr. Pipilis.
Yeah.
Just one short question. So all these pathophysiological mechanisms within the tissue factor comes even before the plaque rupture, because we know that it's the plaque rupture, then tissue factor, and then thrombosis. But this is way back before the...
Exactly. So the monocyte that transforms into macrophage releases tissue factor in the plaque that breaks and produces thrombosis. That's something more or less accepted universally now. Now, yes, by looking into what's happening at the level of the plaque that does not rupture, that was our objective. Let's see if tissue factor is important there. We have found all this function of tissue factor that we can reproduce in vivo by injecting tissue factor in the myocardium. And it's a myocardium, not a plaque. We inject by catheter again. We go and inject tissue factor fragments, because we want to have fragments. And we can see, as we saw in the Hill limb ischemia model, revascularization of ischemic zones. Because when you have a myocardial infarction, you have a piece of your myocardium that is, there is rarefaction. So the microcirculation disappears. You can recover it. So it's another way of looking into tissue factor function. And then we have to go back to the plaque studies, but it's very difficult to study tissue factor in plaques that are still in patients that are alive, because this is the complexity. But nevertheless, my point in my talk today is to highlight the impact of tissue factor in functions that still we don't know, that are going to be important to maintain the physiological homeostasis of the body. And if we understand them, we can use them pharmacologically.
As far as I understood, tissue factor is not found in endothelials, in luminal endothelial cells, so tissue factor is not exposed directly to the blood stream, but is exposed in endothelial microvessels, okay, promotes angiogenesis, and as far as understood, it may be also promote collateralization. Is it correct?
Yes. Yes.
So tissue factor is also involved in collateral circulation, in creating collateral circulation in the myocardium?
Yes.
It was very important.
It is. I think that's why I wanted to speak about this topic. I think this is, well, at least in our hands, we are really excited, and that's why we're going to have mutated tissue factor to avoid, we call it non-coagulant tissue factor, just in case we can evolve this molecule into a treatment to make sure that when we inject this tissue factor, we are not going to produce thrombosis. So we have mutated the factor 7-8 binding spots, and then we now know that we don't induce, we have tested that, we don't induce animals, we don't have any modification of hemostasis. So we are using tissue factor, the part of this molecule that is associated to angiogenesis and not to hemostasis. And this is really important. Tissue factor is a molecule that is, we knew that you require tissue factor for life, because the mouse that were block of tissue factor, they were not be able to be alive. They died in utero in the initial studies done with tissue factor. So tissue factor is important for life. But we have to see, you understand better what else tissue factor does in addition to triggering coagulation and thrombosis.
Thank you very much.
Thank you.
Okay, if there is no other question, we can proceed.